In this video, what I want to talk about is amino acids, and really just a general overview and brief introduction to amino acids, sort of what they are, why they're important, and what to look forward to when we go over all sorts of different things about them. Initially, like I mentioned, I just want to introduce you to them. So what is an amino acid? An amino acid is essentially a compound that has both an amino group with both an amino group and a carboxylic acid group. So an amino acid has an amino group and an acid group. Nothing too crazy about that. Now, why are they important? They're important because they're monomers, right, for proteins. They're monomers, or basically the basic building block for proteins, right? And proteins, we know, are very, very just important in biology, and it's important to understand their structures. So amino acids that we'll be talking about in future videos are going to be alpha amino acids. So what is an alpha amino acid? An alpha amino acid is basically telling you that the amino group is specifically on the alpha carbon. Okay, and that'll make more sense once I actually draw it for you. So this means that the amino group is on or connected to the alpha carbon. Again, I'll explain that in just a moment. Okay, now when I draw these amino acids, I'm going to draw them in what's called the Zwitterion form. Zwitterion or Zwitterion form. And the Zwitterion form is essentially just the form in which it has no net charge. And again, that will make more sense once I've drawn it out. This is assuming at a pH of 7, right, which is a neutral pH. Another thing that we need to know about amino acids is that when we talk about them, we're going to be talking about specifically L-amino acids. And the reason why is because all the naturally occurring proteins, all the naturally occurring amino acids that are in proteins will be L-amino acids. And again, I'll talk about what that means. So all found in L-amino acids in naturally occurring proteins. Okay. So one more thing I want to mention is that all amino acids are chiral. Okay, which is what this L has to do with, right? All are chiral except glycine. Okay. Okay, which is one of the amino acids, one of the 20 amino acids, which is my last point here, is that there are 20 of them. Okay, so these are just some important points to keep in mind. One thing I wanted to do up here is I want to abbreviate amino acids as AAs, not Alcoholics Anonymous, just AAs. Just because I don't want to keep writing amino acids every time I say amino acids. So maybe that would be a little bit easier to follow. And I'll talk about why glycine is an exception, why it's not chiral. So it is not, there's no such thing as L-glycine or D-glycine. There's only glycine, and I'll explain that shortly. So what is the general structure of an amino acid? What does it look like? So an amino acid, well, we mentioned that it has an amino group and a carboxylic acid group, right? So what I'm going to do is I'm going to draw them in what's called the Fischer projection. Okay, so I'm going to draw, actually let me draw it up here. So I'm going to draw the Fischer projection. The Fischer projection. Okay. And I'm also going to draw all these amino acids assuming that the pH is 7. Okay. At a pH of equal to 7, right? Which is why I mentioned this whole Zwitterion form. So an amino acid will look like this. Okay. So here, this intersection point here is a carbon. Up here, we're going to have a carboxylic acid group. Now, because it's an acidic group, right, it will donate its protons. So instead of being protonated here, it's going to look like this. It's going to contain that negative charge. Over here, we're going to draw the amino group. Here we have just an H, and here we have an R group. I want to draw that in blue. Okay, so that is the R group. Now, what are all these different things? So I want to label all of these different things. So this thing up here, right? Whoops, where am I going? There we go. This here is the alpha carbon. Okay, why is it called the alpha carbon? Well, if you remember back to organic chemistry, if you have a particular carbonyl and a carbon right next to that carbonyl will be considered an alpha carbon. So this here, this carboxylic acid group, let me draw it kind of right here just to kind of show you what it would look like. It would look like this. Right? So this is a carbonyl, and then it would be connected to this carbon. This carbon here is this carbon here. So this carbonyl, right, the carbon that is connected to is this carbon. This carbon is considered the alpha carbon. So this carbon, that's the reason it's actually named that. And there are more details in that from organic chemistry, which I won't go over in this video. So this group here, this carboxylic acid group, is considered the alpha carboxyl group. Okay. This here is the alpha amino group. Right, because it's an amino group and it's connected to the alpha carbon. Okay. This H is just the hydrogen. Nothing special about it. It is connected. I mean, I guess you can call it the alpha H, but it's not really of too much importance when we're talking about the amino acids. This R here represents some side chain. Okay. Now, how many different side chains are there? Well, there are 20 different amino acids, so there must be 20 different side chains. Okay, there are 20 different ones. Now, the way I've drawn this here, I've drawn it as an L amino acid. Okay, so this here is an L amino acid. Oh goodness. Okay. Now, the reason why is because in these Fischer projections, we sort any lines that are drawn like this, horizontal, you imagine them protruding out towards you, right? And then any vertical lines, those bonds are going into the plane, away from you. Okay, so if you wanted to change this L amino acid into a D amino acid, well, all you would have to do is take two substituents on that alpha carbon and switch them. So what I normally do is, if I were to draw it, I would just keep the carboxyl group and the side chain in the same locations, and I would just switch the amino group and the hydrogen. Oops, I just said I would switch them and I wrote it again. Sorry about that. So here is going to be the hydrogen, and over here we have the amino group. So when we do that, we change the stereochemistry from L to D. So this would be a D amino acid. But as I said, all naturally occurring proteins have the naturally occurring L amino acids in them. So we don't really sweat the D amino acids. We usually don't draw these out. We don't really care about them. Okay. Now, where do the L and D sort of come from? They come from the basic idea of glyceraldehyde. So I'm going to draw a glyceraldehyde in light blue, turquoise, whatever you want to call it. Now, glyceraldehyde is a carbohydrate. It's a sugar. It looks like this. All right, this is its Fischer projection. Now this one here is D-glyceraldehyde. And this, right, if that's D-glyceraldehyde, you imagine what would happen. Quite simply, I would just switch the H and the OH, so the H would be on the right side and the OH would be on the left side. So this would be L-glyceraldehyde. Okay, so with amino acids, the L form, right? The L form is what naturally occurs. In sugars, the D form is naturally occurring. We'll talk about that later in a future video. Okay, that's just something to kind of keep in mind for now. Okay, but specifically amino acids, L amino acids are the ones that are naturally occurring. Okay, now I mentioned over here that I've already talked about this. What amino acid structure? So hopefully this makes sense, right? Compound that has an amino group and an acid, carboxylic acid group, and this whole alpha carbonyl that sort of explains this alpha amino acid idea. And the Zwitterion form is the whole idea that if it has no net charge, then the carboxylic acid, right, will at a pH of 7, being an acidic group, will donate its proton. The amino group, being basic, will be protonated. It will accept the proton. Again, those definitions, those simple Bronsted definitions, come into play almost all the time. I talked about this. All of them being found in an L stereochemistry. All of them are chiral except glycine. So why haven't I talked about that just yet? So the reason why is because glycine, its side chain, its R group, is just a hydrogen. Okay, so if I drew glycine for you, glycine would look like this. Glycine would have this carboxyl group here, its amino group here. So again, this is assuming a pH of 7, right? Its R group is actually just an H. So what do you notice here? Now, in glycine, what we find is that these two substituents, right, are both just hydrogens, right? So in order to be chiral, you must have, well, I just crossed it out, but in order to be chiral, you have to have 4 different substituents. Glycine does not. Glycine does not have 4 different substituents. Okay, because the R group is just an H, right? So this little dash right next to the R, this little dash right next to the H indicate a covalent bond, right? So this is the covalent bond holding that alpha carbon to that hydrogen. So there are not 4 different substituents, right? Which is a requirement for chirality. Okay, so hopefully that was a good, thorough, brief introduction to amino acids. In future videos, we'll be talking about the actual structures of the different amino acids, how there are some that are polar, some that are non-polar, some that are acidic, basic, neutral. We'll talk about all those different things and kind of ways to remember them. But hopefully this functions as a good introduction to amino acids and how we'll be dealing with them in biochemistry.